Okay, I think we're going to begin. I want to thank you all for coming to the one session of our bioinformatics series. It is a beginning entry session because we are also going to be promoting the um, biomedical informatics boot camp, which is absolutely spectacular and is really, really something that is a great a boot camp that is going to help people a lot with how far you want to go in biomedical informatics with introductory type information. So I just want to say to you, thank you for coming. My name is Catherine Sedota, and I am in the CTSI as the Education Workforce Specialist under Dr. Katya Noyes and Dr. Dave Jacobs. And we have great presenter today, Dr. Peter Elkin, who is the professor and chair within the Jacobs School of Medicine Biomedical Informatics, and he is the person who is chair of the Bioinformatics Department. I also want to tell you that we have two people that are going to be discussing a little bit about the actual core of biomedical informatics, Dr. Samadrala and Dr. Smith, and they're going to tell you a little bit about what the core does. We do have a bunch of workshops. I can put the link in the chat at some time. I just want to tell you that this is the last for this semester, though again, the Biomedical Informatics Bootcamp is going to be going on in July. And we'll put the link also for that if you'd like to sign up for it in the chat. We have a lot of workshops, and biomedical informatics is just one of them. And Dr. Elkin, a couple of years ago, decided, why are we having a separate series when we can do it right at the end of the spring semester? And that can lead you into, in about a month later, the Biomedical Informatics Bootcamp, which is absolutely spectacular. And you can attend these sessions that you think are important to you. So we have done that. If you'd like a certificate of completion, you have to attend today's session. If you don't, we'll have video of it available next week, and there's a short protocol you can do. If you would like the certificate of completion, you also need to attend at least 5 of the workshops within the bootcamp. You either are going to be attending them or you're going to be watching videos of them and writing a short protocol. Either way, I'm going to put this in the chat. If you want to register for the certificate of completion, you're going to contact me, and I'll put my email in the chat. And I'll also put the registration for the BMI Bootcamp in the chat. And you can also attend that, and they're all free. If you'd like CME credits, basically if you're a physician, you answered whether you were a physician, you can get 1.5 credits of CME, and I will be sending that along to the office. And there's going to be a survey I put in the chat at the end. If you would complete that, that is great, because it really helps to allow us to improve our programs, and also we need it for our funding sources. I want to thank you very much. I'm going to stop sharing, and at this time, I am going to turn this over to Dr. Samadrala, and he is in the Department of Biomedical Informatics, and he is one of the people who is the core leader for the CTSI Biomedical Informatics core. So, Dr. Samadrala, on to you.
Great. Thank you, Catherine, for that. I want to second a lot of your comments. Thank you for doing this, Peter. As the expert in biomedical informatics, I figured you should be the one introducing the BMI Bootcamp. But before that, I'll spend about 5 minutes talking about the CTSI Informatics core, which I co-direct. And I'll talk about our part of it, and then I co-direct this with Dr. Smith, and so he can go next. And so our informatics core contributes to CTSI and the CDSA program, which is this NIH-funded program for translational research, by focusing on 3 aims. First, to provide education and support in informatics, and then second, to create an AI-enhanced translational research pipeline. And the third aim is ontology that Barry will talk about. So in terms of the first aim, so our goal is to equip researchers and clinicians and trainees with the necessary informatics tools and expertise to accelerate clinical and translational research. And we achieve this through comprehensive training programs and research support and collaboration and working with people like, we're doing things like the BMI bootcamp and so on, and being part of that. I think Barry and I and Peter, we all give lectures in it. And then there are other things that we do also to provide support. So there's like an informatics support form that if you go to the CTSI website, you can do a service request, and then you can submit any informatics or any bioinformatics problems that you have, biomedical informatics problem that you have, ontology, and so on. And then you can get back some expert advice, initially some consultations, and then you can get help with grants and stuff like that. So that's part of the education and training, but also in terms of the research support and collaboration, we also provide further guidance on utilizing. We have this high performance computing cluster at the CCR. We can give you access to that. We do a lot of electronic health records and analytics. People in the informatics core do that, and so they can provide expertise on this. We have a lot of AI driven informatics tools, and then again we can provide a lot of expertise on that. And our goal is to facilitate interdisciplinary collaboration and ensure that informatics is integrated into research projects to enhance data accessibility and analysis. And so we also work with other members of the CTSA consortium in terms of workforce development, community engagement, and clinical trial recruitment. And we are trying to develop these AI-driven approaches for reducing bias and improving the quality of care for underserved populations. And that's the first aim. That's the research and training aim. And then the second aim is actually doing clinical and translational research. And so we are building out a clinical translational research pipeline that goes from the computer at the keyboard to the bench to the bedside. And that involves this drug discovery platform that we have known as CanDo. And there's a lot of papers published on this, and so you can read about it. But CanDo is a very novel and I would say radical approach to drug discovery, looking at the multi-scale aspect of small molecule behavior and trying to understand those behaviors in how small molecules interact with larger systems in a multi-scale manner. And so we've again, I won't get into the technical details of it, but we've had a lot of advancements. We have several patents, some of which are being licensed and being used by companies to develop drugs. And then we've done a lot of repurposing, so taking existing drugs and using them for new diseases. And then we also have some leads for non-small cell lung cancer and opioid use disorder and aging that we are kind of developing and pursuing. We just disclosed that to the tech transfer office, and we'll see how that goes. So overall, I would say that our informatics core is trying to drive the CTSA mission to accelerate clinical and translational research. And our main way of doing that is by cutting-edge AI-driven platforms, comprehensive education and support, and deep integration with the CTSI consortium. So we're also working on making things drug discovery more accurate and computational drug discovery more accurate, because we think that's where the future is going. And all these AI-driven tools that people are developing, like AlphaFold and so on, would come in very handy to enable us to do this, go from inhibitor discovery to drug discovery. So that's really what the first two aims are. And then I will turn this over to Barry so he can talk about the third aim, the ontology aim, and then we can go to Peter after that. So thank you.
Okay, good. So I'm going to talk briefly about the other half of our informatics core. And so I don't know how many people in the room are familiar with the idea of ontology, but I'll give you a 25-second introduction. And it goes like this. So some of you may have noticed that the Department of Biomedical Informatics here at UB has 5 divisions. And one of them is called the Division of Biomedical Ontology. And so what is biomedical ontology? Well, it's part of biomedical research, which arose in the wake of the Human Genome Project. The problem was that the Human Genome Project was incredibly important to so many people across the world, and they all wanted a piece of the action. And so they started to create genomic and proteomic and toxicoomic silos of their favorite kinds of data. And all of these silos treated the omics data that was being generated in different ways. And that meant that the data was siloed. You couldn't draw together data from Pittsburgh and data from Peking about mice and about rats and draw conclusions because the data were not created in such a way that they worked together. And the solution to that problem was initiated in 1998 by a group of people in a pub in Montreal who represented 4 model organisms: humans, mice, fish, and yeast, I believe. And they decided that they would create a controlled vocabulary for describing different aspects of organism functioning, and they called it the gene ontology. And they did that for various reasons, the result looks like this. This is a tiny fragment of what is now something like 30,000 separate little boxes like this, connected with arrows. This is a part of an ontology. And now I'm a philosopher, and I know something about ontology because it was invented about 2,000 years ago by Aristotle. This is the category system that Aristotle developed. And this category system, or this approach to categories, is still being used by philosophers in all kinds of ways. But it's been discovered by computer scientists. And in a sense, the gene ontology rediscovered what Aristotle was doing with his categories. And Linnaeus followed on from Aristotle. This is a tiny fragment of Linnaeus's ontology of diseases. You can find this fragment in the book edited by Peter on ontology and terminology. I have a paper in there describing the history of biomedical ontology. Now, what happened was that I went to the gene ontology people and I told them about Aristotle, but I also told them that they were making all kinds of mistakes because they didn't know anything about logic or categories or definitions or any of the things that they needed to build a good ontology. And they were convinced and they invited me to help them. And I worked together with the gene ontology to create a suite of ontologies which were designed to work together. So we don't merely have, I'm losing my slides again here. I get excited. They built a suite of ontologies designed to work together. So this is called the Oboe Foundry. It's become very successful. It's used in hundreds of projects over the world. It's growing from year to year. And there are now something like 190 ontologies which are grouped together in this Oboe context. And so what I want to do next, having got this far, what I want to do is I want to extend this Oboe approach, but to include all the ontologies which are being developed in the CTSI here in Buffalo. And because we've been working together with ontologists in other CTSA organizations, we have now quite a number of ontologies. And what we would like to do is to create a consortium, which we already have 8 CTSA hubs who've agreed to join our consortium. They are developing ontologies in tandem with each other. And we're going to focus some of our ontology development work on the field of AI. So everybody wants to use AI in their medical research. And sometimes it works, but sometimes it doesn't work. And we think that we should keep track of when it works, but also keep track of when it doesn't work. And so we're going to develop a very large ontology of aspects or tools or approaches to AI which are used in medicine or in biomedicine or in healthcare. And then we're going to use that ontology to tag data which we receive from our ontology consortium members according to which type of AI program was being used, how it was being used, and whether it was successful or failed. And in this way, we believe we can save people time in using AI approaches which are destined to fail and point them in the direction of AI approaches which are destined to succeed. And I think that's probably enough.
Great. Thank you so much. I appreciate it.
Thank you.
You can stop sharing, and then Dr. Elkin, if you'd like to put your slides on the screen. And Dr. Elkin will be giving me his slides at some time after the presentation. So when I do send everyone the survey tomorrow, even though I'm going to put it in the chat, you could do it today if you wanted to. You're going to get the slides then. And
Sorry,
then Dr. Elkin, if you'd like to put your slides up.
All right. Are you seeing it properly?
We are. You are all set to go. Thank you so much. And I'll turn it over to you. Dr. Elkin is the chair of the Biomedical Informatics Department, also professor of internal medicine, surgery, orthopedics. He wears many, many hats, and we're very pleased to have him today. Thank you. I'll pass it over to you.
Thank you, Catherine. What we're going to talk about a little bit is a field in medicine called biomedical informatics. And I'm going to do it in the context of our department, and then we'll talk about quite a bit of the research that we're doing. And then also I have some parts of the presentation that require audience response. So I am a clinician. I'm a physician who practices still both inpatient and outpatient. And in addition, I have an active research program and do both training and administration. In our department, we have 5 different divisions. There's the Division of Clinical Informatics. There's the Division of Bioinformatics, which Dr. Samadrala chairs or is the chief of. We have Public Health Informatics. We have Biomedical Ontology, and Werner Kustis is our chief of Biomedical Ontology. And then we have Sociotechnical and Human-Centered Design. And those deal with things like human factors engineering. Here are some of our primary investigators in the department. It's quite a good list of people, including Ram and Barry that you see here. And then we've graduated, some of these are graduated fellows, but these are some of our fellows that we've had over the years, and some are now on faculty. We have quite a number of PhD students in the department, and we also have a good support staff with a number of programmer analysts. And in addition, we have an assistant chair, and we have Gloria, who holds us all together. And, we're looking to fill a position for an educational coordinator. And we have a lot of adjunct professors who work with us. Some are actually full-time with the department, but it's just troublesome to move them from other places to here. So we use them in that fashion. That our vision in biomedical informatics is the global impact through biomedical informatics innovation, and to continue to be a top 10% of medical schools ranked biomedical informatics program in the nation, using our innovation to improve the global health of our patients and their families. So we have a new specialty called clinical informatics, which is a subspecialty of any other primary specialty in medicine. It's approved by ABMS, the American Board of Medical Specialties. And people who train in this as physicians go on to become chief medical information officers, like the ones we have at Kaleida, Christian Federick, who's just come here from Pittsburgh. And I'll let the fellows tell you about this themselves.
Hello, my name is Dr. Peter Elkin. I'm the program director for the Clinical Informatics Fellowship at the University of Buffalo. We have an outstanding range of cutting-edge research, very strong administrative experiences for our fellows, where they sit in the boardrooms of our hospital systems, along with the CEOs, CFOs, and CMIOs. We have a wonderful teaching experience with an outstanding faculty, and fellows are able to get their master's degree should they want to while they're doing their fellowship. We have a wonderful camaraderie between the fellows and the faculty, and a wonderful set of opportunities that will prepare you for every facet of clinical informatics practice.
Everybody in the program at the University of Buffalo Clinical Informatics is incredibly supportive. Any projects that you're interested in getting involved with, you'll meet with the program director, discuss your unique interests and goals, and then synthesize a plan over the course of 2 years as to how you can meet and achieve your objectives and goals. It prepares you very well to take the board exam when you're finished.
My interest in the program lies in the fact that we have such a wide range of areas of study, which includes public health informatics, pathology informatics, clinical informatics, and bioinformatics. In addition to that, we have a large group of outstanding informaticians who have an open door policy. They're always willing to help.
We rotated several of the local hospitals and one of the best regional health information exchanges in the country. So far as a first year fellow, I've been able to work on projects that are analyzing COVID-19 patient data, reviewing physician alerts within the EMR. I'm currently working on a project with the Regional Health Information Change to look at population health data.
One of the projects that I was involved with at ECMC was a project that looked at data migration and data management. The hospital system was moving towards a new EHR in the ambulatory clinic setting, and this involved almost 20 different clinical sites. One of the issues that came up during one of the meetings was that of data migration. We weren't able to migrate all of the clinical data, so we had to figure out a way to define data migration on behalf of the hospital and all of the different specialties. I worked directly with the CMIO, worked with end users, attended executive leadership management meetings, and eventually figured out how, based on participatory design, we could develop a concise list of data endpoints based on feedback from unique specialties. It was a great project to be involved with, taught me a lot about project management, and it's something that I developed skills that I'll take with me in my future in clinical informatics.
One of the projects that I am currently working on and I'm very excited about is looking at the impact of COVID-19 on telemedicine training in undergraduate medical education. Now, although telemedicine has been around for a few decades, with COVID-19 knocking at our front doors, we've all had to readjust. This includes medical schools having to readjust their curriculum to include telemedicine in training future doctors to use technology more effectively for improving patient outcomes.
Buffalo is a fantastic place to live. It feels like a big city, but everybody's very, very friendly. There's shopping, restaurants, nightlife, wonderful outdoor activities, hiking along the gorge, the Niagara River, and we're right next to Canada.
Now, if that sounds exciting to you, and you're a person who likes to take on projects, then the University of Buffalo Clinical Informatics program is probably for you, and we would love to see you here.
Should you have any questions, please do not hesitate to contact me at elkinp@buffalo.edu. We look forward to seeing you here in the spring.
Thanks. We really like that one. So we have a lot of educational experiences in the department. We have a medical student rotation that rotates through with our fellows in the hospitals. We have a set of research rotations, so trainees like residents and fellows can come and do research with us. We have a master's degree in biomedical informatics, which we offer. We also have a PhD in biomedical informatics, and that includes MD, PhD candidates. We have an ACGME accredited fellowship in clinical informatics. We have postdoctoral fellows in that are training in science through an NLM grant, and we also have a short-term training experiences over the summer where undergraduate master students can come and do some research with us. And then we have an undergraduate major that we started, and we're graduating our third class. It's our fifth class that we have entering. And then we are now a thread in the medical school across all of the medical school curriculum, which I think is great and is really an exciting new change to teach people how to do practice medicine in the 21st century. And recently I've been thinking about the kinds of EHR competencies that we need to test medical students on in order to be licensed to practice. And I think this is really important because it really is a patient safety issue and something that we're pursuing. Our grant from the National Library of Medicine is very prestigious. We're one of only 18 sites in the country that have this grant, and we've had 4 additional supplements to it to do new things, and we are considered by the National Library of Medicine one of the top sites in the country. And we have this R25 program. It actually provides $5,000 in payment for the students who work with us. So if you know an undergraduate or a master's student that would benefit from that next year, although we're full this year, let us know. And then there's $3,000 housing allowance for people from out of town. They all do the summer boot camp. Here's a picture of one of our classes. Everybody looks very happy. And this is our master's program that we have, which is done usually over 4 semesters, but you can do it over 3 if you want to be fast. We also have a joint program with the School of Pharmacy in a master's in Biomedical Informatics and PharmD program. That's a 1-year out program, and it goes through the summer. And then we have a PhD program, which is a little heavier than most. It has about 10 courses in it rather than 5 in most PhD programs. We have an active grant-funded program here in the department where people are getting quite a bit of funding. I wanted to tell you a little bit about the field and some of the things I think are the better accomplishments of the field, so you understand why we think we've contributed considerably. One is precision or personalized medicine. The mapping of the microsomal enzyme oxidase cytochromes to the changes that need to be made in drug prescribing based on the specific genetic code of the individuals was done in combination with pharmacists. And this was codified into RX norm, and that's how it gets into clinical practice in most decision support systems. E-prescribing. So it used to be, if you guys remember, that there was a joke about doctors' handwriting because prescriptions were so hard to read by the pharmacist that errors were very common in prescribing meds. Now it's all done electronically through NCP2P script, which we developed with the pharmacy community as well, and that's how all prescriptions go into pharmacies now, and how we monitor better what people are taking. Evidence-based medicine couldn't have happened without mesh codes and the ability to do systematic reviews and meta-analyses, and this all came from the work of informaticians at the National Library of Medicine. And then clinical decision support, it's not where it should be yet, but it certainly is helpful. And this is something that physicians come to rely on in their clinical practice and helps them to practice safe and effective medicine. Electronic health record systems, it's not they're not where they should be either, but everything that's in them came out of the laboratory originally. We're implementing Epic, which I guess Bob Wachter said is the cream of the crop. I know the Epic system, it's getting better every year, and I'm looking forward to having that in Buffalo. Translational research. So this funding that comes in through the CTSA came from a program by the National Center for Advancing Translational Sciences. It turns out that this was started by informaticians. So in the year 2000, Andy Ballas published a book chapter that said it took 17 years to go from bench to bedside. In 2001, this was a paper with Elizabeth Borden, and this got Congress talking. They wondered why, when we give so much money to science, it was taking so long to get these things into practice. And by 2005, the largest grant program that the NIH had ever given, the CTSA program, was inaugurated. This was all due to Andy's efforts, and he's still a colleague of mine, and we do research together, and I was on the phone with him just today. And I can tell you that the mark that he's made is significant. It now takes 14 years to go from bench to bedside. You might not think that so much, but it helps it. Each year that we delay in getting things to the patients that actually work, is life-saving for many, and we actually do better. But we need to do better than we're doing now. And in 2009, the NIH created NCATS, the National Center for Advancing Translational Sciences. And so we've been, in the biomedical informatics community, very effective in moving science forward. Mapping the human genome. In 2003, Bill Clinton got up and said, "We have mapped the human genome." Did we map everybody's genome? No. Who did we map? Well, it was one guy from Buffalo, who was an African-American guy, who Craig Ventnor had gotten the blood from when he was here on staff faculty. And that person's blood is in the 38th year of the reference genome that we all use in every single genetic application that we have. So Buffalo's had a huge impact on molecular biology. And then systems that facilitate paying for physicians. It used to be that we paid bad doctors more than good doctors because they'd hurt the patient and they get paid again for fixing it. And the person who did the right thing in the first place just got paid once. And also, and so this in pay for performance, which was done by informatics because we figured out how to measure the quality of care that physicians give to their patients. This in part was some of my work that went into this. And the idea is that by doing so, we can actually risk adjust people and populations, and that the insurance companies can pay people more for taking care of sicker people. And if they do well on the patients that were sicker to begin with, they get paid even more. And then we created PubMed. PubMed is where you find the medical literature. Without that, people couldn't practice medicine properly and wouldn't be able to do research. Research would grind to a halt in the country. There were 12 hours a month ago when PubMed was down, and people from the whole world were screaming about it. And that just shows you the impact of the National Library of Medicine and of the medical literature in allowing people to practice good medicine and for science. But what the most important thing that we're doing for you personally is systematizing the practice of medicine, taking it from a cottage industry where you practice the way you were trained into a systematized practice of care. And this is incredibly important for the future of effective and safe medical practice. So I'm a doctor, so I'll tell you, this is a case. This is Bill Knievel, who's a 49-year-old male on vacation with his family in Florida. His driving conditions are wet and slippery, and after being cut off, his car impacts an embankment. Unconscious, he's taken to Florida General Hospital's emergency department. He routinely gets his care at St. Elsewhere in Boston. Records are requested by Florida General Hospital, and text records are sent which show Mr. Knievel to have a past medical history of hypertension and type 2 diabetes. He's currently taking lisinopril, hydrochlorothiazide, metformin, and aspirin. In the ED, he's found to have an MI with a troponin T of 0.24, which is elevated. His CBC was normal. He had a creatinine of 2.7, which is elevated, as normal electrolytes, and a glucose of 187. His beta-hydroxybutyrate was not elevated, and his bicarb was 24. Some people confuse that with the ketoacidosis, but it isn't. He had ketones in his urine. Mr. Knievel's continued on what happens presently is he's continued on his present medicines. A beta blocker started. After 2 days, his serum potassium 7.2, which is elevated. His bicarb drops to 14. His rhythm degenerates into ventricular tachycardia, followed by a cardiac arrest. And in spite of an aggressive resuscitation attempt, he passes away. What do we expect to happen in the future? Well, data from St. Elsewhere is transmitted to Florida General Hospital in a standards-based codified interoperable format using ontology. As you heard from Dr. Smith, the doctor overworked. The admitting physician uses an order entry system that provides decision support and incorporates into its logic engine the data that was transferred from St. Elsewhere. The system alerts the admitting physician that with a creatinine of 2.7, the metformin should be discontinued due to the serious risk of metabolic acidosis. Doctor overworked. He sees the metformin and puts him on sliding scale insulin instead. 2 days later, Mr. Knievel's potassium's normal at 4.7, his bicarb's 24, which is normal, and his creatinine is 1.7, trending down to his baseline of 1.3. And on the fourth hospital day, he's discharged in good condition, and his family decides to take a repeat trip to Disney World next year when Dad feels a little better. So for Mr. Knievel, informatics standards and Florida General Hospital's clinical decision support system made an immeasurable difference for him and his family. Just a more modern example. Michael, Mr. Knievel's son, is inconsolable. His pediatric records from St. Elsewhere show that he's considered healthy, but due to his behavior and separation from his parents, he's visited by a child psychiatrist. It's determined that Michael's suffering from an acute stress reaction. They wish to prescribe him a selective serotonin reuptake inhibitor, namely escitalopram, which is the brand name Lexapro. However, Michael's St. Elsewhere records show that his DNA sequence data to have a polymorphism associated with a non-functioning CYP3A4 enzyme, indicating that Michael would be a very poor metabolizer of escitalopram. Therefore, he's placed on sertraline or Zoloft, which is metabolized by the P450, CYP2D6, and 2C19 pathways, where he's known to be a star1 extensive metabolizer phenotype. This medication decision avoids subjecting Michael to potentially serious medication side effects that may have occurred through inadvertently overdosing him with escitalopram. And this just shows you that every patient that we treat is worthy of our absolute best effort. And this is an example of where decision support can be helpful in putting guardrails around clinical practice that keep our patients safe, and we prescribe effective but safe medications. So we published a paper, and this was published in Nature Communications Biology, where we found that RNA editing had a big role in human genetic disease. And this was a way that we improved people's knowledge of what molecular biology is, and will, in the long run, lead to new treatments for patients. And this was published just last year. So how much should a clinician be expected to remember, and how much should the computer be able to provide decision support to his or her practice? So there are 6,000 articles published every week. I read 60 last week. I got 5,940 behind. Thousands of ongoing clinical trials. The average clinical guideline is about 67 pages long. Average new patient visits, 30 minutes, with follow-ups are 15. Some are maybe 45 and 30, but it's not more than that. And the average elderly patient has 6 active clinical problems. With my practice, it's 23. And clinicians make hundreds of decisions a week. So what level of medical error and harm is it acceptable to expect from a practicing clinician? What do you guys think? I gave you some choices here. Someone want to take a stab at this?
You can either unmute or you can put it in chat. So what level of medical error and harm is it acceptable to accept from a practicing clinician? Your choices are 1, 2, 3, 4, 5, and 6. So please answer something. If not, please put it in chat.
Yeah, if they
Yes, it's okay.
Would you monitor the chat, Catherine?
I am.
I can't see it. And if someone does that, let me know.
No problem.
Otherwise, I'm happy to call on people.
I have someone wrote number 3.
So what is that? The medical error or harm?
That's 0.01%. Other people are putting 1%. There are question marks after it, but a lot of people are putting 1%. So 0.1 is one answer. 1% is the overwhelming answer at this time.
Okay, so the rate of medical error currently is actually 6%, and this is on multiple studies. Not only that, but it was independent of whether it's a community hospital or an academic hospital. It was exactly the same number. And it turns out that 17% of these go on to cause harm. So 1% of everything a doctor does leads to harm. 99% to the good, 1% to the harm. And or if you think about the error, 94% to the good and 1% to the harm. And in fact, that's unacceptable. If you were getting on an airplane and I told you you had a 1% chance of crashing, you would turn around and get right off that plane. You wouldn't take that risk. But you go in the hospital, and that's the exact risk you have when you go in the hospital. And so we need to do better. We need to take it to Six Sigma accuracy, like flying a plane, which is parts per million. So we need to do that. In order to do that, we need to make use of all our data. We need to increase the clinical decision support and support best practice medicine, not just minimal standards of care. And we need integrated clinical decision support. That includes diagnostic workup, treatment and protocols, and outcome surveillance. And we need improved communications pathways, messages to and from you need them the way you need them, because that's how people think these days. And we published a paper with Gillian Franklin, one of our faculty in the department, as the first author. And this was done in the journal Life. And this is about biases in machine learning algorithms. And there are lots of types of biases in machine learning algorithms. Most people think about algorithmic bias in it, but there are many other types of bias that we have to guard against. And if you're interested in this topic, this is a great article. I really recommend it. And we use ontology. That Barry actually was one of the founders of the basic formal ontology, and many scientific ontologies have revolved around that. And I have worked with him and Werner Kustis on some of their ontology projects. And they use and advocate for the ontology of general medical sciences as the to take care of courses of illness in the representation of the knowledge. And then we use SNOMED, LOINC, and RxNorm as the detailed ontologies underneath it. And then we do one thing that nobody else does. We build them into sentences so that we have semantic networks of these relationships that come from our both our authoritative knowledge and our clinical understanding. And we've actually trained a set of knowledge embeddings and knowledge graph embeddings and also knowledge and graph embeddings in order to teach machine learning algorithms, AI, and to teach them. We kind of send our AI to medical school. The first thing we do is teach it how we speak in medicine. Then we add in all of what we know about medicine, including pathology. We include the general literature, all the clinical guidelines, all the guidelines for patients. We included all the drug data in the country, and we included all of the toxicogenomic database, and then many other things like order sets and discharge checklists and any other authoritative piece of information we could find about clinical care. And then we asked the question, well before there were LLMs, if we have a machine learning algorithm that actually has these pre-trained embeddings, so we actually have baseline knowledge. It's more like a doctor when it sees an x-ray or a doctor looks at a piece of a clinical case. They have their whole training to fall back on and put the case in the context of what they know about medicine. And we thought machine learning algorithms should have the same advantage. So this is a project we did where we looked at opioid overdoses and death events in a population of opioid using people, about 170,000 people from the state of New York. And we found that it was much more accurate when you had the pre-trained medical knowledge, and that the ROC curve for it was better, and that we found, interestingly, that it was less biased. So the lines at the top are compared with the lines at the bottom, which are with and without the pre-trained embeddings. And you can see that the bottom ones without that degrade more quickly as we either remove people from the cohort, or we remove data randomly from the cohort, or we add back bad data to the cohort. And still, in every which way, it performed better with the pre-trained embeddings. Then large language models hit the scene, and we did this experiment, which we published in JAMA Network. And this is an AI pipeline that uses our large language model along with the llama models, and it takes questions from the three United States medical licensing exams. The first one you take during your second year of medical school, the second one during your fourth year of medical school, and the third one your internship. So they get progressively more clinical. And basically, we take our NLP, high definition NLP, and we parse it. And this creates the semantic net. and then we put it through the LLM, and that finds all the knowledge that we know about that's associated with the facts that are found in the question or potential answers to the case. And that allows us to increase the amount of information that goes into the LLM. That focuses its attention and gives us better answers. Again, here's the case. This is a 24-year-old recent college graduate who comes to the office from because of a 3-year history of increased frequency of urination and occasional incontinence whenever she travels. She says the urinary symptoms typically begin a few days before she's going to fly, and they stop the day after she arrives at her destination. She says she is anxious about flying, and she reports having similar but milder symptoms prior to exams before graduating from school. Her history is otherwise unremarkable. Her medication is only oral contraceptives. Her vital signs are normal. Physical exam has no abnormalities. Urinalysis and urine culture are normal. And so these are the potential answers that the USMLE question gives you. And the native LLM gets it wrong and says reassure the patient that her symptoms will resolve in time. And the semantic clinical AI, or SKY, we put in all these facts that we've learned, about 44 kilobytes of facts, and then it actually gets the right answer. It says recommend behavioral therapy because the person has an anxiety reaction. And here's the results of the study, which show that Sky was definitely better in all models than without it, and that when you start to get to the 70 billion parameter model, although the 405 billion parameter model was a little better, it wasn't statistically significantly so. And so you start to see that it's starting to plateau. And so, people who say that the bigger, just all we need are bigger models and it'll get all of knowledge is not correct. I think we're going to chew through what we know is authoritative. And then after that, we're going to start getting misinformation and advertise the information and other things that are going to negatively affect the large language model's ability to perform. And so we need new kinds of reasoning. And if you talk to Google and others, that's what they say is the next step in this. And this is the first that I know of publication about a new method of reasoning beyond simple chain of thought reasoning, which is used by LLMs today. This is our chatbot. It's available to you now, and you can find it right here. And if you click this little box that says Semantic Knowledge Reasoner, then you get Sky. And if you don't use it, you see what the native 70 billion parameter llama model would tell you for the same question. Put the questions in right here, and the answers come out here. And here's the little checkbox. This is the Gardner Hype Cycle, and for those of you who are older and were around in 1991 when this AI first got developed, we're now up here in this hype cycle, and we're trying not to get to what we call the valley of death down here. And so what we need in order to bridge that curve into excellent and working systems is really strong evidence trials, so randomized prospective controlled trials that show the efficacy that can bring it into practice the same way we would a medicine or other device. And we've already got 500 approved AI devices by the FDA, and I think we're headed toward 10,000 by 2030. So, what question thing didn't come up? Okay, never mind. Let's go on to that. Back. Okay, so I want to tell you about some of the research in the department. We're doing some aging research, and we think we may have found at least one of the main triggers or controllers for aging in the population. People have been looking for the fountain of youth forever, and we think we may have found a way to reverse aging. And so we took a database called GTEx, which is a database of normal people, and we compared people in their 20s, 30s, 40s with 40s, 30s, 40s, 50s, 60s, and 70s. And we looked for things that were progressively downregulated or progressively upregulated. And we looked at it in pathways that are related to telomere growth or length, which is known as a marker for aging. And we think this could be a potential intervention. We did this in many different cell types in single-cell RNA-seq, and we were able to actually find that furin regulates the Notch pathways. And what we did was we looked back in the cascade until we didn't find this pattern. And then we set up some wet lab experiments. And what you see is that this happens consistently across tissues, but this was done in skin cells, muscle cells, macrophages, and neurons. And then we went to the lab and we have younger cells here, and we look at older cells, and this is the amount of furin that was in them. And you can see that that and the Notch pathways, at least Notch 2 and Notch 3, were downregulated. And then we did something pretty miraculous, we think. We took the young cells and we looked at their ability to form new muscle. And then we looked at the older cells, and they couldn't do it anymore. And then we transfected those cells with a plasmid for furin, and look at it, it actually came back, and these were functional muscles that contract. So we were able to reverse sarcopenia in aged muscle cells. And this is a model that's been well tested in the literature. And this was all done within 5 days. Now, obviously, with more treatment, we may get better results, but within 5 days, we had very significant regrowth. And when we looked at the fusion index, it actually was better than the young cells after 5 days. People don't generally do aging research because they think it'll take years and years and years, and we were able to reverse aging in days. And yes, of course, we've put in the provisional patent and are doing additional experiments on human tissue. This was a mouse model. And then we looked at the breakdown products from this. So, when you have dysfunction, often what gets you is that other things in the pathway will build up and they cause dysfunction of the cell. And here, GZMB and HES1 were the two things that built up. And after we transfected the aged cells, it knocked it down to close to normal. So we think that we've solved a lot of the problems that are coming up in aged cells. And then we wanted to see how this would work in a disease such as Alzheimer's disease. So we looked at the pathways and we found that furin causes atom 10 and taste to decrease, and this causes an increase in beta amyloid, which is what builds up in Alzheimer's disease. So we believe that this will actually be a treatment for mild to moderate Alzheimer's disease as well. And if you've ever seen an Alzheimer's patient, you know how important that discovering a treatment for that would be that actually works. So we decided years ago that we wanted to cure cancer. And we had a bunch of other studies that we did preliminary to this, but we published a study recently in the Journal of Cardiovascular Surgery, which is a high-impact journal, showing that the addition of metformin to stage 1 non-small cell lung cancer led to an 18% survival advantage. Now, that's better than most really high pay-for drugs that you pay $30,000 a month for. Metformin's like $5 a month, and it can actually improve survival. The current survival for stage 1 non-small cell lung cancer is 47%. And so you can see that it's a tough disease. And even with surgery, it's not perfect outcomes. So we can do better. And this is one of the ways that we can do better. And then we decided to try to look at why that was so. So we went to the laboratory and we took cell lines of both adeno and squamous carcinoma, and we grew them in successively less sugar. And of course, with less sugar, they grew more slowly. And then we added back ketones, which is when you go on a low-carb diet, what you start to burn. And people are able to burn and live off ketones perfectly nicely without any trouble. As a matter of fact, within 6 weeks of a low-carb diet, you are completely running off of ketones. You're not running off sugar anymore. And yet the cell cultures were not rescued by the ketones. They were not rescued by it. And so we found that this may be the difference. And the whole idea of treating cancer is to win the race against cancer. You want our host defenses to be able to take care of the cancer faster than the cancer can grow. So if we slow the rate of growth in cancer, we may win the war. Thank you. What we're trying to get toward really is a learning health system. And in order to create a learning health system, we need more of these kinds of AI tools in order to do faster implementation science and get things implemented toward precision medicine in the practice and continue to learn. I am chairing the best practices group at NCATS for learning health systems, and we're hopefully going to come out with some good recommendations for all CTSAs. Please send your best work to the Journal of Translational Research. I'm the editor-in-chief. We really would appreciate your best work. This is a Taylor and Francis journal. And Barry already plugged our book, but if you're interested in ontology, after Barry's talk, our book is a perfectly great place to go to get it. It's one of the Springer Nature series books, and we really I should say all of the authors did a great job in this book, and I'm very proud of it. And this is the boot camp that Catherine talked about earlier. And here's the schedule for the boot camp. You can find it at the URL that's there, or you can just put your camera up and get the QR code. And that should let you just get on. I'll leave that up for a second. You can see there's many different lectures in it. I think I do the most lectures, which is 4, but there's a ton of people, and I think most everything is on here. There may be one additional lecture that didn't make the slide that came on late. And I would encourage you to share this with anybody you want. It's free. It uses Jupyter Notebooks on our Jupyter Hub, and gives you real hands-on experience in coding. And you learn Python, you learn R, RStudio, you learn all kinds of things. You don't have to take every lecture, but take the ones that you think are right. And as Catherine said, you can get a micro-credential or a digital badge, I forget which it is, if you take 5 of these. And the idea is they should be in in an area of concentration. You can see that there's all of that in there. And if you really love it and you want to do our master's degree or something, please do get in touch with me. We would be thrilled to have you.
And this is for a certificate of completion. Just want to make sure everybody knows.
Oh, but
Okay.
we were working for a digital badge or a micro.
There is a digital badge that we have that requires courses to be taken. So if you do want to discuss one that doesn't require courses to be taken, I can be happy to meet with you and we can see what we can get together with that. So totally up to you.
Yeah, we are happy to offer that too, and with our master's courses, but we certainly want people to be well trained who come out of it. So it represents our educational product. It represents a strong individual. So I'm just going to give you a couple of quotes that I've found interesting over the years. One is by Machiavelli from 1505, who said, "There's nothing more difficult to take in hand or more perilous to conduct or more uncertain in its success than to take the lead in the introduction of a new order of things, because the innovator has for enemies all those who've done well under the old conditions, and only lukewarm defenders in those who may do well under the new." And that is just as true today as it was back in 1505. So that's sort of an amazing longevity for a quote to be relevant. My favorite quote is by Peter Drucker from Harvard, who said, "The best way to predict the future is to create it. Come create the future with us." We're excited to have you. And with that, I thank you for your kind attention, and I'll stop sharing and keep my screen open and happy to answer any questions anybody might have.
Great. I do have some questions that were actually sent to me, so I'm going to start with the ones that are sent to me. If you have any other questions, please type them in the chat or you can unmute. I'm just going to make sure I get the 3 questions that were sent to me so that you can answer them. The first one is, for clinical faculty who have access to large data sets for research but lack funding support, what resources, internal or external, are recommended to assist with data cleaning and analysis?
So I think for the data cleaning, people can collaborate with us, and that means that if their papers are on the papers, and if there's grants, we're on the grants, and people have to be willing to commit to that in the department. The CTSA itself offers about 10 hours of biostatistics free service that I believe the biostatistics core is running, and I'm pretty sure that's still active. It was the case, and that's where I think you should go for those services. For the data cleaning, that's not usually something biostatisticians can do for you. It's usually something the informatics community can do for you. And so I would talk with Dr. Samadrala about that.
Excellent. I'll go on to the second question. For faculty members aiming to enhance their skills in R for more advanced data analysis, what learning opportunities or resources would you recommend? And are there specific courses, workshops, or programs at UB that you suggest? This is Michael Sellis. So if he is here and he wants to unmute, perhaps he can address that even further.
Yeah, so Michael, I don't know how strong you are in R already, but there are several courses in the boot camp on R and R Studio that would be worth it. And I think there's 2 or 3, and there's also in research methods as well that could be helpful with that. And other than that, we have a research method classes that we teach in our master's program that also teaches skills. And there's a number of the different courses in our biomedical informatics masters that do teach R&R Studio skills in a practical way. We also teach structured query language for getting the data, and we teach data cleaning, and also that data cleaning and data basically all the security and not only data cleaning but all the different data wrangling methods are taught within the boot camp, but also are part of our coursework within the master's program. And we'd be happy, if you're interested, to talk to you more about that.
Yeah, no, thank you very much. This has been extremely helpful. And I think I'm newer to UB. I was at Damon previously. So I'm just really trying to understand my resources. Like, do I use the Bird Core in the biostatistics department or the bioinformatics? But just looking to get assistance. And I am a clinical faculty member. So it's typically not grant based, but just looking for collaborators that can assist with large data sets of patient data.
Yeah, so there's like I said, there's some curated data sets that we already have within our department, and then uh, there's if you're looking at claims data, the biostatistics department's a really fair place to go for that. If you're looking for EHR data, then probably coming to the biomedical informatics group is probably a better place to spend your time. And and I'm glad to I'm glad to meet with you, but we also have others in the department that would be perfectly willing to meet with you and talk about your needs.
Perfect. Thank you very much.
Great. And then Alice has a question. Will you be approaching biopharmaceutical companies in the AI and research compilation?
Oh yeah. We're already doing that. I mean, I work with Pharma Foundation regularly. I'm a part of their translational research group and everything. So yeah, we're very and I'm on the board of ACTS, the Association of Clinical and Translational Sciences, and I'm on the AMIA board, and I'm also vice president of the Friends of the NLM. So I know most of the people in science that do this kind of work.
Great. Thank you. Those are the questions I had. If anybody else has any questions, please feel free to unmute, or also you can put it in the chat and I will read it for you. So please do it either of those ways.
I just have one follow-up question from my first question. You did mention the 10 hours of biostatistics service. Just clarifying, is that through the Bird Core, the BERD core, or is that
Yes.
through a separate entity?
That's through the Bird Core.
Okay, thank you.
You're welcome.
Anyone else? If there are no questions, we want to thank you for your attendance. If everyone's done, go ahead. Go outside and have a nice day.